Hey everyone! So today, we are diving into something that is literally changing how we see life itself. We're talking about a completely new way to figure out the shape of proteins. And the secret sauce is, well, it's pretty incredible. It's about treating biology less like chemistry and more like a language.
So, let's just start with a huge question. What if we could predict the 3D shape of pretty much any protein almost instantly just by looking at its basic code? I mean, what would that even unlock for science, for medicine, for everything?
You know, for decades, this has been one of the holy grails in biology. Figuring out a protein's 3D structure is absolutely essential. It tells you what it does, how it works, which is how you design new drugs or understand diseases. But actually finding that structure has always been this incredibly slow and difficult puzzle. Okay, so to really get why this is such a breakthrough, we have to look at how things used to be done. For the longest time, even our best tools were stuck behind this major roadblock, something we can call the alignment bottleneck.
And that bottleneck has a name: multiple sequence alignment, or MSA for short. The basic idea was, okay, if you want to know the shape of your protein, you go out and find thousands of other similar proteins that we already have the structure for. The thinking was, hey, if they're related from an evolutionary standpoint, they probably fold in pretty similar ways.
And when you break it down, you see just how much of a slog this was. First, you have to search these massive databases to find those related sequences. Then, you have to painstakingly line them all up to find the shared evolutionary clues. And only then could a model even start to guess at the 3D structure. It worked, sure, but man was it slow and computationally expensive.
And that right there, that is the bottleneck. This massive brute force search and compare job, this hunt for clues across thousands of different sequences, that's what's been holding back protein prediction for years.
But what if you could just skip that whole step? This is where a massive breakthrough from Meta AI just completely changes the game. They came up with a new way to do this that just sidesteps the bottleneck entirely by thinking about the problem in a totally new way.
So let's just put this side by side, because it makes it super clear. On one side, you've got the old way, comparing one protein to thousands of its relatives. And on the other side, the new way, predicting a protein's structure directly from its own single sequence. No big family reunion needed.
And this right here is the magic of it all. The model, which is called ESMFold, treats that string of amino acids, the stuff that makes up a protein, like it's a sentence. It's not just a chemical formula, it's actually learning the grammar of biology, the underlying rules that dictate how that sentence is supposed to fold into a complex working machine. So how'd they do it? Well, Meta AI released ESMFold back in 2022. It's built on what's called a transformer architecture. Yep, the exact same kind of tech behind large language models you've heard about. And to teach it the language of biology, they fed it a mind-boggling amount of data, 250 million protein sequences.
Okay, so it's a whole new approach. That's cool. But so what? Why does this actually matter in the real world? Well, it all comes down to one thing: speed. We're talking blazing fast, game-changing speed.
I mean, just look at this. By cutting out that massive alignment search, ESMFold can predict structures orders of magnitude faster than the old ways. We're talking minutes instead of hours, or sometimes even days. The difference is just, it's staggering. And that speed creates this incredible ripple effect. Yeah, you get faster results and it costs less, but the truly revolutionary part? It can run on a regular consumer-grade GPU. This completely democratizes the field. It puts this power into the hands of smaller labs, educators, anyone who doesn't have a supercomputer. And it's an absolute game changer for studying brand new proteins that don't have any known relatives to compare against.
So this newfound speed isn't just some theory. It's not just some cool lab experiment. It has already been put to work to create something monumental, a brand new atlas of the biological world. Over 600 million. Let that number sink in for a second. That's how many protein structures have been predicted with this tech and just released to the world in an open source database. This is a gift to science. It's revealing the shapes of proteins from microbes we've never even seen before. It's like we're getting our first real look at the dark matter of the biological universe.
When you step back, what this really is, it's this beautiful convergence. It's a moment where the tools we built to understand human language, natural language processing, were turned around and used to decode the fundamental language of life itself.
And that leaves us with this incredible final question to chew on. For the very first time, we're really becoming fluent in the language of proteins. We can look at a sequence and almost instantly understand what it means in 3D. So, what secrets are we going to unlock next? What are we going to find?